So this talk is going to be a little special because it's going to talk about childhood memories and so personal relationships, and then about work which we have done in the footsteps of Timofeeff. So I showed you this. I can't move my slides now. I don't know what to do with this. Okay. I showed you this slide already in the morning. I don't know how many of you were there. This is Timofeeff-Ressovsky at young age at the Kaiser Wilhelm Institute for Brain Research in Berlin-Buch, now Max-Delbrück-Center. And he arrived there at the age of 25 years. And my father, who was a biophysicist, also a director of a Kaiser Wilhelm Institute in Frankfurt, knew him actually, and they were friends. And not only this, but also when we look inside this greenhouse where Timofeeff is standing here, you can see a lady in the middle. And the lady in the middle is a lady, she died unfortunately recently, Natasha Komm. She was my aunt, the sister of my mother. And she worked with Timofeeff for the entire 20 years he was working in Buch, and was full of stories about him. So then there was this connection, which I also showed this morning, between Timofeeff and Max-Delbrück, documented in this famous paper. And that connection was somehow also important for my own career, because when I started my work, I was working at the Genetics Institute in Cologne, which was actually founded. I'm sorry, this is. Which was actually founded by Max-Delbrück. And so I knew Max-Delbrück from those times because he came every year visiting, and sometimes we talked about our genetics work. And our genetics work at that time was imprinted by several of the people who are here on this slide. But one important figure here was a person called Hua Gu, a Chinese student actually, who developed during the time in Cologne a technique which is called conditional gene targeting by using Cre-LoxP recombination. And at that time, together with collaboration with Jamie Mars in Vancouver, we developed this this system where one has LoxP flanked target genes in a mouse, which can be then deleted in a cell type specific way by Cre transgenic mice, of which lots of them were generated over the coming years. And the whole new era of research became open and accessible, really quite along the lines of what Timofeeff-Ressovsky had started to do in Berlin-Buch, using mostly Drosophila as a system. I then went for 10 years to Boston, to Harvard Medical School. This is a picture of my group there. And after these 10 years, I returned to the Max-Delbrück-Center for Molecular Medicine in Berlin. So back to Timofeeff's roots. And this is my group at that time. And here Here is what I showed this morning, the entrance to the present Max-Delbrück-Center with this Torhaus here, which was a place where Timofeeff lived at the time when he was working in Buch. And I've shown you in the morning this Timofeeff's bust here in front of the Timofeeff house, a big lab building in the center of the Berlin campus, reminding all of us of this founding great spirit in loco. So after this the excursion, I want to come to work we have done in Berlin-Buch since that time, and I'll focus on work which concerns the area most closely related to Timofeeff-Ressovsky's work, namely work on gene targeting and gene repair in somatic mouse and human cells, as it can be done nowadays in the times of conditional gene targeting, and then of course CRISPR-Cas mutagenesis. And I will introduce you a little bit into the first topic I want to talk about, which is Epstein-Barr virus related, by pointing out a few basic facts about Epstein-Barr virus. It's a human tumor virus, which is with which we are almost all of us latently infected. And it is a virus which transforms human B cells, human antibody producing cells, into malignant cells. And we would all die from these infections. But fortunately, our immune system, in particular T cells and NK cells, recognize the EBV infected cells in a process which is called immune surveillance, and the immune system kills these cells off. And somehow 1 in a million cells survives for a lifetime, and the immune system is constantly controlling these infected cells from further expanding. Now, for the transformation of the B cells, there are 2 proteins which are particularly important virus proteins, which are called latent membrane protein 2A and latent membrane protein 1. The latter most important for the transformation. And these molecules are using signaling pathways used by B cells naturally, by mimicking certain molecules involved in the signal transducing chain in B cells, which is the B cell receptor for antigen and CD40 co-receptor. And these are constitutively acting proteins, so they activate the B cells and drive them into proliferation. So when this became clear, we tried immediately, with the help of conditional gene targeting, to model this in mice. And the amazing thing that one can model this very nicely. One can put the LMPs as transgenes into the mice, make them inducible by the Cree protein, ER fusion protein, which can be activated by tamoxifen. And so by giving these mice tamoxifen, one can remove the stop cassette and express the LMPs in B cells. And what happens, you can see here in the lower part of the graph, 6 days after tamoxifen induction, the B cells in the animal begin to dramatically expand. And they would kill the animals, as you can see on the right-hand side of this graph, except that when the T cells in the mice, exactly like in the humans, they attack these infected cells and eliminate them completely from the animal, and the animals survive. So that is a mouse system of acute EBV infection, which we have here in our hands. And then we use this the system to model and to cure, develop therapies for fatal inherited lymphoproliferative diseases, which block T cell and NK cell killing, and are therefore making in humans the affected people susceptible to EBV and other viral infections, so that the patients die usually early in childhood. And I'll concentrate on one particular form of these diseases, that is perforin deficiency. It's called familial hemophagocytic lymphohistiocytosis type 2, perforin deficiency, which is one of these diseases which is fatal for young children. And here's what happens in our mouse model when we take perforin deficient mice and induce in these mice LMP1 expression by tamoxifen, by the system I just showed you. The animals will all die, shown in these red curves, whereas the controlled mice, as I showed you, which are perforin proficient, survive. And you can see that that in these perforin deficient mice on the right-hand side, there is this liver which is completely whitish, and is full of tumor cells. And one can actually see by flow cytometry that in the perforin deficient mice now the B cells expand uninhibited. They also expand in the liver of the animals, as shown on the right-hand side, and kill the mice. And this is in this system, the perforin deficient mice mimic perfectly the human disease in many symptoms like liver failure, anemia, hepatosplenomegaly, and the acute disease. So the plan was to try to repair in T cells from these animals the gene, and then after repair by CRISPR-Cas mutagenesis now, the most recent revolution in this field, to inject back the repaired cells and see whether we can rescue or protect these mice. And then, of course, the idea would be to continue with this approach in the human. And, finally, I want to switch to a last system we are doing a lot of work on, actually, in the lab with many different ramifications. And that is to try to repair genes instead of in T cells, mature lymphoid cells, in hematopoietic stem progenitor cells. And the plan would be, of course, in the mice and in humans, to isolate from the patient's hematopoietic stem cells, which can be easily done from blood and bone marrow, then do gene correction, and then transplant the cells back to the human. That's, of course, done now in many laboratories all through the world. I just want to show you a few slides on one particular disease where we've tried this out and shown how very efficiently this approach presently works. And that's a disease which is called severe congenital neutropenia. These are patients who cannot generate neutrophils, which is a very severe condition. It's fatal, actually. And most of these patients, 55%, fail to do that because of mutations in a gene which is called ELANE gene, neutrophil elastase. And in these conditions, the final maturation step of the cells from progenitor cells through promyelocytes to neutrophils is blocked completely. So we had discovered we had gotten cells from a patient who had a particular new mutation shown here on the lower part in the hematopoietic stem cells, and we got some vials of hematopoietic stem cells from bone marrow cells, one would say, from that patient. And the two people in my lab who worked on this, Wantung Chu and Tung, we're establishing a repair system for this. And this is just showing how it is done in some detail. I don't want to go into the details. It's a classical way of doing it by providing a template which is repaired. And then we have two different ways to repair this. We can repair either only this specific mutation or in principle every mutation in this particular exon. And this work is published, so you can look at the details if you wish. And when we do this, we get again by isolating hematopoietic stem cells, electroporating them, putting in ribonucleoprotein with Cas9, and then the donor template, we get efficiencies of up to 30% of repair in the hematopoietic stem cells. And when we induce in these stem cells from the patients in the repaired stem cells the neutrophil differentiation in vitro, we get on the right-hand side here this almost the same frequency of neutrophils as we get from healthy donor controls here on the left side. And this is accompanied by neutrophil elastase expression. And we can also show functionally that these repaired neutrophils are now forming the famous nets, DNA nets, by which they capture bacteria. That is shown here in the upper panel for the repaired cells as compared to the controls. And in the lower panel, you can also see that these repaired neutrophils are now killing bacteria in vitro assay as efficiently as do wild-type neutrophils. And finally, we can show by transplantation of these cells into humanized mice that the cells can be transplanted in vitro, repopulate the animals over long periods of time, and are essentially ready for clinical trials if adapted to GMP conditions. I think that's really what I wanted to show you. I'm sure that Timofeeff would have been happy to see this. It's done all over the world now. It's a wonderful perspective. This is Wantung Chu. This is Tung Tran. And this is again Hyun Lee, who did this work in the Epstein-Barr virus model. And then I have just a slide with lots of collaborators, grant support, and I stop it here and will be happy to answer questions. Thank you.